Hi everyone, and sorry for the slight delay. Technical problems, which same could probably be said for parts of Andy's PhD as well, mostly induced by myself. But before I begin, I'd like to begin by acknowledging the traditional owners of the lands on which we're meeting here today, the Wurundjeri people of the Kulin Nation, and acknowledge and extend my respects to their elders past, present and emerging, and to any other First Nations people joining us today. Well, today we have a very special seminar, and not just because at the moment the only people in the world who are going to be able to listen to it are the people in here. So 2019, doesn't it feel? So this is the group that matters. I can say anything I want about the online group. Ooh, I don't know if we're live for that. Well, anyway, so I'm really excited to introduce Dr. Andrew Baldi to present today. Andrew was one of the first people to join my lab a few years ago, and he's an amazing guy. So, in his spare time, he's a paediatric hematologist, which means he treats children with cancer, looks after kids with bleeding disorders, and he puts all that in his back pocket and then joins us and works on global health, and particularly on developing and learning a whole lot of new skills around advanced genomics and genetics as applied to samples from the field. And he's done an incredible project, well, I hope you'll agree, where he's literally combined being out there in the villages, collecting samples from in remote parts of rural Bangladesh. Along with that, training our field team in long distance running. So he has improved the fitness of our team over there, and taken them on every morning on early morning runs all over the villages of rural Bangladesh, and he's brought that all back here, and I think you'll see done something quite unique, using cutting-edge techniques available here at WEHI to address some of the most important problems that face children in low-income countries. And I'm really looking forward to hearing your talk, and I hope you'll enjoy it. Andrew.
Thank you, Santa, for that introduction. So my talk today centres on the intestinal microbiome and as it relates to global child health based on a field study in Bangladesh. I put this slide here at the beginning, not because I thought I would forget, but because I just really wanted to emphasise how pivotal this team in Bangladesh was to this project and this research, in particular in the photo on the left, Dr. Jenna Hamadani, as well as Dr. Imrul Hassan. This was a monumental undertaking, collecting biological samples in a field in a rural part of Bangladesh, a low-income country, where it's very important to maintain the cold chain and all of those things, and transporting during COVID. And so these highly skilled team really were central to everything that I present to you today. So I just wanted to begin by just, I guess, noting that a child growing up in a low and middle income country faces a number of threats to his or her health and healthy development. My research centres on how the gut microbiome plays a role in interacting with those public health, with those child health challenges. Before I zoom into the gut microbiome, I was just going to walk you through some of these child health challenges rather briefly. But here I've nominated anemia, stunting, infectious disease, and antibiotic use and antimicrobial resistance as 4 of the key challenges facing children in low and middle income countries today. So firstly, anemia is defined as a reduction in the circulating red cell mass and hemoglobin such that it's insufficient to meet the oxygen demands of the tissues of the body. And it's a massive problem, particularly in children under 5. Around 270 million children worldwide have anemia. And you can see from this map that the key populations are in sub-Saharan Africa and in South Asia. The causes of anemia are multifactorial, but around half is attributable to iron deficiency. And this is such a priority because it is linked to impaired growth and development of children. And it's such a priority that the World Health Organization recommends universal iron interventions to children, whether in the form of iron syrup or iron-containing micronutrient powders, to children where the background prevalence of anemia is high. So this is done without necessarily a clinical examination or laboratory diagnosis of anemia, and with the knowledge that not all anemia is caused by iron deficiency. And in this setting, clinical follow-up is rare. Turning to stunting. So stunting is defined as a failure of linear growth. So it's height that's more than 2 standard deviations below the WHO child growth standards for age. So here you can see the centile chart with age on the x-axis and the z-scores. And again, the causes are multifactorial and multi-sectoral. It's a consequence of inadequate nutrition, but also recurrent infection. And interestingly, it's even more complex than that because nutritional interventions don't achieve the outcomes that you would expect as well. So there's more to it, and there's a lot more to be learned from more research in children with stunting in these areas. And again, the worldwide prevalence of stunting is centred in sub-Saharan Africa, South and Southeast Asia, with a global prevalence of 22%. But you can see from this map that there are areas in the 30 and 40%. And again, it's such a priority, similar to anemia, that there are global nutrition targets developed by the World Health Assembly. So by 2025, one of these targets relates to stunting, and it is to achieve at least a 40% reduction in the number of children under 5 who are stunted. And this figure shows that 53 countries in the world are on track to achieve that target, and Bangladesh is making good gains in this area. And just briefly on infectious diseases, we know that infection is the leading cause of mortality in children under 5 globally, from diarrheal disease, from pneumonia and malaria. Actually, the global under 5 mortality rate declined considerably in recent decades. However, diarrheal disease is a leading contributor to death from infection, and not only is it due to the high infection burden, potentially from poorer sanitation and reduced hygiene practices, but access to healthcare is a major factor because this group of diseases is largely very treatable. So antibiotics are often deployed to combat these infections, but paired with that is the threat of antimicrobial resistance. And again, Southeast Asia and South Asia are considered regions of very high concern in the growth and emergence of resistant bacteria. And a recent estimate says that states that 4.95 million deaths have been associated with bacterial AMR in 2019. And again, the causes are numerous, including the misuse and the overuse of antibiotics, but also the infection burden and the need for antibiotics, as well as the use of poor antibiotic practices in agriculture, for example. But one of the key components of this causation model is the availability of antibiotics without a prescription and without appropriate indications and without proper stewardship. So this map shows areas in the world where the percentage of antibiotic use that's conducted without a prescription, and you can see Bangladesh there next to India, that 86% of all antibiotics used were done so without a prescription. So these distinct areas of global child health are vitally important to a child's survival and also their ability to achieve their growth and developmental potential. And my research here henceforth is on the role of the gut microbiome in these domains. So I use the term microbiota to describe the microorganisms within a niche, so in this case in the gut, and the term microbiome to refer to the organisms as well as their other genes and their functions and their metabolism. So going back to these domains, firstly, with regard to iron, there have been several studies in children, mainly in sub-Saharan Africa, that suggest that iron may reprofile the gut microbiome in a potentially pathogenic manner. So iron in this study of Kenyan infants who were randomised to receive a micronutrient powder either with or without iron showed that there was a reduction in commensal bacteria and an increase in enteropathogens. So you can see on the figure on the right, these are box plots showing log fold changes in different bacterial groups. So in yellow is the enteropathogen Escherichia shigella, and on the right is the potentially commensal bifidobacterium. And you can see the rightmost colour of each box shows the intervention with iron. So you can see an increase from baseline to end of intervention with iron for Escherichia and a decrease in bifidobacterium. So, and a systematic review has suggested a modest increase in diarrhea also related to iron supplementation. And so this is important. So if you're reprofiling the gut microbiome and potentially leading to an increase in diarrhea, your intervention for good may be achieving harm. There has been some research in childhood stunting as it relates to the gut microbiome, and a lot of the research shows that that children who are stunted have a different microbiome profile compared to their non-stunted peers. So a study in Madagascar showed that there was a decompartmentalization of the microbiome, so with oral bacteria being present in the gut microbiome, as well as a higher abundance of enteropathogens. And similarly, a small study of children in Bangladesh associated abundance of different species with length for age. So you can see on this heat map, these were all children under 2 years old. But for example, on the top right, children under 12 months, LFA is length for age on that right column. You can see that with a higher length for age, there was a higher abundance of commensal bifidobacterium in the yellow. And with a higher length for age, there was a relative reduction in the enteropathogens. But there are still there's still a lot to learn about the microbiome and its role in stunting, particularly because we know that dietary interventions aren't as effective as we would like them to be. And the gut microbiome is also known to be a source of antibiotic resistance. It's an ideal reservoir for the development of antibiotic resistance. It's host to a high number of very diverse microorganisms and bacteria that are living in close proximity under different selection pressures, not just from antibiotics, but for nutrition, and finding their particular niche. And so it's known that antibiotic resistance can develop and can be transferred horizontally to other bacteria. And this is a photo showing how readily available the antibiotics are in this area of Bangladesh. Readily cheap and readily available from a stall just near the field laboratory. So there's potentially a very complex interaction between these factors in global child health and the gut microbiome. So for example, diet can directly lead to poor diet can directly lead to anemia, but it can also potentially be modulated through the alterations that may be caused by environmental contamination, by iron supplementation, and leading to inflammation and then malabsorption of nutrients. So you can get to anemia from that perspective as well, even with an adequate diet, and you can also and also the altered intestinal microbiome is implicated in antibiotic resistance. So I wanted us to turn to what questions this raises. So the first is an obvious one: is it safe to give iron supplementation to children in these settings, or are the harms unacceptable? Do the harms exist, and are they acceptable? And what are the interactions between the microbiome and childhood stunting, and how can we harness some of that knowledge to develop effective interventions to improve growth of children? And what are the effects of antibiotics on the microbiome in children and on AMR, especially in regions with a high prevalence of infection as well as antibiotic use? And this is a very busy ward in the diarrheal hospital in the capital, Dhaka, that sees many patients every day. So turning to the BRISC trial, this is the trial that I leveraged to develop the microbiome study. So BRISC is the benefits and risks of iron interventions in children. So, the setting was rural Bangladesh, a couple of hours outside of the capital Dhaka. So Bangladesh is the 8th most populated country in the world, with 169 million people and a high number of people in rural areas. It has a stunting prevalence of 28% in under 5s and a high incidence of anemia in this same age group of at least 43%. So just briefly, the BRISC trial was an efficacy trial of iron in young children. So children were recruited at the age of 8 months and received a 3-month intervention. So it was one of iron syrup, iron in a micronutrient powder versus placebo for the 3 months. And iron reduced anemia at the end of that intervention, and it also improved iron stores, but interestingly had no effect on the primary outcome of the trial, which was cognitive development. And also, interestingly, there was no increase in diarrhea. So iron was effective in correcting anemia and iron status, but had no effect on childhood development after a 3-month intervention. This was published in the New England Journal of Medicine in 2021. So we thought this was an ideal setting because of the high prevalence of anemia as well as stunting, to study the role of the gut microbiome in those two public health domains. However, at the beginning of the study, it was we were struck by, with these visits at recruitment and during the weekly visits during the trial, there was a strikingly high amount of antibiotic use. So during this 13-week intervention, around 43% of children had at least one antibiotic dose, and 28% had 2 courses of antibiotics during that intervention. So actually, I used this as a retrospective cohort as well to study the effects of antibiotics on the microbiome and on AMR. So the hypotheses based for this cohort were, and based on the previous research, were that iron would lead to a reduction in commensal bacteria, such as bifidobacterium, and an increase in the relative abundance of the enteropathogens that I discussed earlier. With regard to stunting, my hypothesis was that there would be important differences in the microbiome between stunted and non-stunted children, with a higher abundance of enteropathogens in those with stunting, and that antibiotic use would be associated with a decline in microbial diversity, and that AMR genes may be enriched in the samples from children who'd received antibiotics. And this is one of the field workers giving a nappy, a reusable nappy, to a mother of one of the children on the trial. So this happened many hundreds of times during the trial, and the field workers also, were very dedicated in once the sample was obtained and put in a jar by the mother or the father, going out on their motorbike to get it, no matter what time of day or night it was, to take it straight to the lab, so then it could be processed and frozen pretty quickly. So this was the sub-study design. So all children were recruited at the age of 8 months. They received the intervention or placebo for 3 months, which took them to an 11-month sampling time point. And then the BRISC trial itself had a follow-up time point 9 months later, when the children were 20 months old. So the stool sampling time points were 8 months, 11 months, end of intervention, as well as a follow-up time point. And the way I carried out the sub-study was to recruit the last 1,000 of the 3,300 children that were on the main BRISC trial. So as I said, parents were given nappies, which were greatly appreciated, because most children weren't defecating into nappies. And then the nappies had liners, and also mothers were given a specimen jar. That specimen jar was taken straight to the lab. Parents were given the mobile number of the field workers, so there would be no delays in having the samples processed. Here's a photo of one of the workers aliquoting the samples. An aliquot went to a stool alone that was frozen, and one had DNA preservative added to it. And then they were put in a -20 in the field, and then transported to the -80 in Dhaka before they were sent to Melbourne. So a very enormous undertaking by these workers, and I'm very appreciative to them. So the approach that I took to the workflow, to the lab work, was to perform 16S amplicon sequencing on samples, and on a subset of those samples, shotgun metagenomics. So 16S, the 16S gene is a gene common to all bacteria. It contains some constant regions, but also some hypervariable regions, and it's those hypervariable regions that allow you to identify the bacteria, usually down to the genus level. And it provides good taxonomic information about what's in a sample, and can be done relatively cheaply with modern sequencing techniques. Shotgun metagenomics is a method of fragmenting the entire metagenome of a sample, and that allows you, with reference libraries, to get down to the species or even strain level. And it also, because you're sequencing the whole metagenome, you can also obtain other information about the genes that are present, so you can get some functional information, but the cost is much higher. So my approach was to perform 16S on the bulk of the samples to get a big sample size, and then with the shotgun to perform that on a subset of samples to achieve a higher resolution to get that functional information. So with 16S, I'm largely going to be presenting the taxonomic profiling as well as the diversity measures, and with the shotgun, the functional analysis, particularly when I'm talking about antibiotic resistance. So just a couple of definitions briefly. So when we talk about diversity, we're often talking about alpha diversity and beta diversity. So alpha diversity, the figure at the top there, is biodiversity within a sample or within a community. There are a number of indices that can be used. So you can see the community on the left has a higher alpha diversity because there's more of a variety of different coloured circles there. But the Shannon index and the inverse Simpson index are commonly used because it's not just about the number of taxa that are present, but it's also about their distribution and whether one's more dominant or whether they're evenly distributed. And those indices consider those factors in different ways. And beta diversity is more dealing with similarities and differences between samples or groups of samples. So you can see what they have in common and whether they cluster together on a PCOA plot, for example. So just turning to what I obtained. So I obtained 2,281 samples that had good 16S sequences and just under 950 shotgun samples. These were evenly distributed by intervention arm. And my priority for the 16S samples was to get as many as I could. So the way that the workflow ended up going was that I had more baseline and that end of intervention time point, which I'll be mainly presenting here. In terms of the participant characteristics, there were no major differences with or biases with sex or the village the child was from. And children had quite a high adherence to the iron or placebo of around 78% had a high adherence above 70% over that 13 weeks. Baseline anemia was 44% and iron deficiency 28%, and the combination of iron deficiency and anemia was 20% of children, and 23.5% were stunted at baseline. So just first, I just wanted to talk about the changes that occur with age, because this is by far the most, the biggest source of variation in the microbiome that we see. So at birth, the gut microbiome is predominantly dominated by bifidobacteria, particularly in breastfed babies, because bifidobacteria is able to metabolize the oligosaccharides present in breast milk. And then over time, there's an overall increase in diversity. And then the microbiome tends to resemble an adult profile by the age of 2 or 3. So what I saw, just because I had these longitudinal samples between the ages of 8 to 20 months, and this is 16S analysis of alpha diversity here, you can see by Shannon index and Simpson index, the inverse Simpson index, that with age there was a kind of a stepwise increase in alpha diversity, which is in keeping with what has been previously reported and what we know. And there was a clear clustering using beta diversity analysis on this PCOA plot between the samples from the youngest and the oldest children. So the green shows the children who are 8 months, and the blue 20 months, and you can see those from a particular age have a lot more common with each other than they do with the older or younger samples. These box plots show at the top the 16S data and at the bottom the shotgun data next to each other. So you can kind of see at the phylum level the compositional changes that take place in terms of the changes in relative abundance at those 3 time points, those 3 ages. So, for example, you can see on the left the actinobacteria phylum, which contains bifidobacteria, has a relatively high, or has a high relative abundance in the youngest children, and there's a stepwise decrease with age in both the 16S and shotgun data, and also in proteobacteria, which is the 4th one along, which contains a lot of those enteropathogens, and they're known to be higher in neonates and young infants. And there's also a decrease in them over time, which is what I would expect. In fact, all of these changes are statistically significant, apart from the very modest changes in the Firmicutes in that 3-month time period that I've circled, that I've got a red square around. So turning to the effects of iron interventions on the microbiome. So again, my hypothesis was that iron would lead to a reduction in commensal bacteria and an increase in the abundance of those enteropathogens. But firstly, I just wanted to show you these forest plots from the main trial that showed that that the interventions were effective. So you can see here that iron versus placebo and MNPs, the micronutrient powders, versus placebo reduced the anemia prevalence. And they also improved iron status. So you can see the prevalence rate of iron deficiency is reduced considerably. And although there was a trend, there was no significant increase in diarrhea after giving iron and MNPs compared to placebo. So interestingly, there was no change in alpha diversity with iron. So here is the alpha diversity plots again with placebo, iron and MNPs on the x-axis. So you can see no significant changes at that 11-month time point, so immediately at the end of the intervention. I then wanted to look at, similar to previous studies that have taken place, look at differential abundance at different taxonomic levels. So my approach to this was to do this in a hierarchical fashion and do it based on hypothesis. Based on the literature and also on biological mechanisms, I pre-selected 6 genera to perform an initial unadjusted analysis, and I did this with 16S and shotgun using several methods. And then I followed this by an exploratory analysis that was adjusted for multiple comparisons, so of all the genera and species present. And because some of the iron literature doesn't adjust for multiple comparisons in their analysis, and I wanted to have a hypothesis-driven approach. So these are the genera that I selected and the reason why. So it's mainly based on the reported research. So Bifidobacterium and Lactobacillus are 2 key genera that contain many commensal species that have previously been shown to reduce in abundance with iron. Escherichia and Enterobacter are 2 examples of the potentially pathogenic Enterobacteria. And in that previous study in Kenya, Escherichia was increased with iron. And Enterococcus and Prevotella. Enterococcus is a commensal, generally a commensal, but is also an opportunistic pathogen known to change in abundance with iron. And Prevotella also has been known to be lower in non-anemic infants from a previous study, but iron is also known to stimulate its growth. So this volcano plot shows, based on that first analysis of the pre-selected genera, that there were no differentially abundant genera found with iron or MNPs, so all are below the line. So this is log-fold change on the x-axis and the negative log 10 of the p-value on the y. looking at all the genera present in the sample with this exploratory analysis. On the left is unadjusted and on the right is adjusted. So there are no significantly differentially expressed genera with the adjusted analysis. And those that were above, that were above the red line there on the unadjusted analysis were no longer significant after adjustment for multiple comparisons. Looking with the shotgun data down to the species level, but again, just from those 6 genera, there were no differentially abundant species detected. And again, on the exploratory analysis at the species level, there were no differentially abundant species from the remainder of the data set. So in summary, the iron intervention was not associated with any significant changes to the gut microbiota, which was not what was expected, especially given that obviously the iron and the MMPs were efficacious in improving anemia. So then I wanted to look at the interaction between microbiome and stunting. So firstly, I told you that 23.5% of children were stunted at baseline, and the prevalence of this increased from 8 to 20 months, and it is known in global child health that the prevalence of stunting kind of peaks between 18 to 24 months, particularly where there are in areas where there are poorer sources of complementary food after breastfeeding ceases. So there are no differences, again, in alpha diversity between stunted children and their non-stunted peers at that baseline time point at 8 months of age. So again, you can see no significant change on Shannon or inverse Simpson indices, and no differentially abundant species or genera between the stunted and non-stunted children at that baseline time point. And this was true for the species level and at the genus level. And it was true for all of the time points. Then I wanted to take a broader look as well at the phylum level. And interestingly, at this same time point, there were considerable differences in the relative abundance of two very prevalent phyla. So again, the actinobacteria was shown here to have a lower abundance in the children who were stunted. So that's the phylum that contains bifidobacterium and a lot of the commensal species. And conversely, proteobacteria, which contains the family of enteropathogenic bacteria, was increased at this time point, and significantly so. For the children later on, at 20 months, there was a trend towards the same, but this wasn't statistically significant. The BRICS trial also collected data via some surveys about food security, and there's a standard food security questionnaire that's used in these situations about access to food, whether families have had to go without food or skip meals, and the quality of food that's available to them. So overall, 80% of the population surveyed was food secure, but importantly, 20% had one of mild, moderate, or severe food insecurity. And you can see I just plotted food security and versus food insecurity for those 2 phyla that were differentially abundant with stunting status, and there was no association between food security and the abundance of the actinobacteria or the proteobacteria. So what we could glean from the data here was that there is a clear relationship between stunting and food insecurity, but there's clearly some more complexity here, and we need to kind of have a properly designed trial, prospective trial, to examine this in more detail. So in summary, with stunting in 8-month-old children, there was a higher relative abundance of proteobacteria and a lower relative abundance of actinobacteria. So turning to the antibiotic use and the microbiome, here's another stall where the antibiotics are readily available. So I hypothesized that antibiotic use would be associated with a decline in microbial diversity. And just to show this figure again, 43% of participants had used at least one antibiotic during the intervention period. And so with that knowledge, again, I use this not in an RCT style, but as a retrospective cohort, because we had the samples from baseline and we had the samples immediately at the end of intervention. So I wanted to look at the effect of recent antibiotic use. So I define that as antibiotic use in that final week prior to sampling, in a slightly earlier time definition of weeks 9 to 12. And then also what the effect would be, if any, of earlier antibiotic use in the first 4 weeks only. And you can see antibiotic use was relatively similar from week to week. So the indications for antibiotic use in this cohort were largely fever and upper respiratory tract symptoms. And so the vast majority were administered for that reason. And diarrhea was lower down. And interestingly, concerningly, the classes of antibiotic that were given or that were used during the intervention period were pretty heavy-duty antibiotics that often in our high-income setting are much less available in the community. Cephalosporins are used commonly here, but there were some cephalosporins that would not be available in Australia that were used among these children. And the macrolides, chiefly azithromycin, where azithromycin was very commonly used by children here, where here in Australia, because of our antimicrobial stewardship and strict prescribing restrictions, it's reserved for very particular circumstances and indications. So antibiotic use in that final week leading up to sampling was associated with a drop in alpha diversity on those two indices, which is in line with what I had hypothesized. However, even going back just 1 week to that earlier time point, there were no significant differences, and that decline wasn't seen, nor was it seen at that earlier definition of antibiotic use, just in the first 4 weeks. So I wanted to examine that final week in more detail. So looking at the differential abundance in an adjusted analysis at the genus level using the 16S data, you can see there was an increased abundance of Escherichia shigella, so that member of proteobacteria in the enteropathogen family with antibiotics compared to those who had no antibiotics at all. And looking at the species level with the shotgun data, you can see there was a significant increase in 2 species of Enterococcus, so Enterococcus faecium and Enterococcus gallinarum with in that final week. With those earlier definitions, there were no significant changes in differential abundance. So with that antibiotic use late, nor with the antibiotic use in the first 4 weeks. So it really was just that final week in the lead up to that sample time point. So there was a reduction in alpha diversity and an increase in those two in Escherichia on the 16S and with Enterococcus on the shotgun. And here's another antibiotic store, but I think this is a more of a formal pharmacy. So I'm just going to turn to antimicrobial resistance now. So with the hypothesis that AMR genes may be detected in the samples from children who received antibiotics. So to perform this analysis, I used the CARD database, the Comprehensive Antibiotic Resistance Database from McMaster University. So it has a host of reference sequences that are arranged into antibiotic resistance ontology, or ARO, which is kind of the organizational unit. So it allows you to have genes or several genes grouped into an ARO that doesn't depend on which species they're from. There's a marker set developed by the biobakery team that I used to run the shotgun samples through. So firstly, and this has been shown in some previous studies, that overall abundance and carriage of these antimicrobial resistance genes is highest in early infancy and reduces with age. The previous studies that are of note often look at neonates in a hospital setting, but this was shown to still be the case between the 8-month time point and the 20-month time point. So and the y-axis is the abundance of the ARO reads. And interestingly, the overall abundance of ARO, so this is regardless of antibiotic use or not, there's a pretty strong correlation between just the overall burden or carriage of these AROs and the relative abundance of Escherichia, you can see in the red in this scatter plot, and also the phylum that Escherichia comes from, Proteobacteria, in the blue, and to a lesser extent, Enterococcus on the left. So, there appears to be, in terms of just overall antimicrobial resistance gene abundance, a strong association between that and the underlying taxonomy of the sample, regardless of antibiotic use. So this is, again, a volcano plot showing just in that final week of antibiotic use that there was an association between antibiotic use here and an increase in several of these AROs, you can see in the red. And this is an adjusted analysis. And from the reference database, the CARD database, these are all actually related to Enterococcus faecium, which you'll recall is one of the species that was increased in abundance with antibiotic use. The particular ARO descriptions or mechanisms relate to target alteration of the antibiotic or inactivation of the antibiotic, but are not. There's no association between the drug class listed here and the drug classes that were used in among the cohort here. And there's no significant increase in the ARO carriage with earlier antibiotic use in weeks 9 to 12 or antibiotic use in the first 4 weeks of the intervention. So, in summary, the effects of antibiotic use on antimicrobial resistance that we saw here only were confined to just that week prior to sampling, with use of antibiotics in that final week associated with increased AROs here related to Enterococcus faecium. So in summary, firstly, iron interventions are not associated with significant changes in the gut microbiome diversity or any significant changes in the composition of the microbiome in these children. Stunted infants have a lower abundance of actinobacteria and a higher abundance of proteobacteria than their non-stunted peers at that earliest time point, so potentially more pathogenic microbiome, and this operates independently of food security. With regard to antibiotic use, there was a very strikingly high rate of antibiotic use in this group, and it appeared to lead to a transient reduction in the microbiome diversity and an increase in Escherichia shigella, as well as Enterococcus. And as I said, the changes appear to be transient. And finally, antimicrobial resistance genes were detected at high levels among children who received antibiotics in that final week. They were related to Enterococcus faecium, and they were not increased in the samples from children who received antibiotics earlier. And also, AMR gene abundance correlated strongly with the underlying abundance of Proteobacteria and Escherichia. So the value of this study was that it really examined a population in South Asia in detail. Previous studies relating to iron and the microbiome, as I mentioned, were centred on children in Africa, and this was also had a large sample size that utilised the randomised control trial model as well, with a placebo, which many previous studies lack. The use of both 16S and shotgun metagenomics allowed me to cover a great breadth of the cohort, but also using shotgun to have a more higher resolution analysis and a functional analysis for the AMR. And it is indeed one of the first and largest studies to investigate the effects of community antibiotics on AMR. The vast majority of the literature and the research to date has been in high-income countries and dealing with hospital prescription of antibiotics. So my concluding remarks are that universal iron interventions are safe in this population with regard to the gut microbiome, and that there's a lot more to be learnt about the relationship between the pathogenic microbiome and stunting in the first year of life. And based on this kind of retrospective look at the cohort, that community use of antibiotics induces transient changes in the gut microbiome, and other mechanisms do need to be explored. So finally, I would just like to acknowledge everyone who helped with this trial, and I'd particularly like to acknowledge both the NHMRC for their assistance with this trial, as well as the Gyokha Wong Charitable Trust for providing some laboratory support for some of the analysis in Melbourne. I'd like to thank Santa and the rest of the Pasricha Laboratory for all of their assistance, particularly in helping a medical doctor learn some lab techniques. Seems like a long time ago now, and it was. And particular thanks again to the team in Bangladesh for their tireless work and dedication and their expertise. Thank you.
Thanks, Andy. And as you can all see, we now have a lab uniform. So are there any questions from the floor? I can't see any online. I don't know if I've got the wrong thing, but yeah, I'll start with Marnie, although I'm meant to be carrying microphones around. I should probably be down there.
Thank you. Oh.
Now it might work. Yeah, okay. Great seminar. So for the third time. I wonder, have you had a look at any of your metrics, like stunting, for example, rather than making it yes or no, but using the data you have, which might be able to go think of it as a spectrum? And would you gain any more power by doing that to look at your correlations?
Yeah, potentially. This is not the last that I will be working, the final time I'll be working with this data set. But yeah, we have the raw measurements and the length for age Z scores. We also have data on wasting and underweight status of children. So I haven't looked. I've looked specifically initially at length for age, and also from an initial analysis that I looked at the change in the length from 8 to 20 months and whether that baseline microbiome was predictive of how a child would grow in that year, and there didn't seem to be a close correlation. But yeah, there's a lot more analysis that I can do with that.
Great talk, very interesting study. I'm wondering, maybe like, do you think whether and how the maternal factors, like for example the food insecurity, can also happen in the mother? What do you think the maternal factors can impact the microbiome of the child? Or do you think these factors can imprint already the microbiome change or the iron deficiency before the child will be born? So do you think there would be some correlation?
Yeah. It's an excellent question, and the answer is yes. So antenatal and kind of the immediate postnatal factors have a huge role to play in particularly early life growth and iron status. I think a previous study of stunting was looking at causative factors, for example, and showed that birth weight is a really key determinant, which is primarily a maternal factor, and we know that that iron status and kind of that kind of iron health begins kind of prenatally as well. So we're a bit limited. So actually 8 months of age is relatively old for an infant. Yeah. So we have information about birth mode, like cesarean versus vaginal delivery as well, which is known to affect the microbiome of neonates and young infants. I haven't looked at that separately in this age group, but yeah, the maternal health is definitely a big factor.
Hi, Andrew. Thanks for that talk. That was really interesting. I was just wondering, with your, I thought it was really interesting that you didn't see sort of any differences in the microbiome with the iron supplements. And I was just wondering, with the 8 to 11 months of iron supplement, is that like standard practice, or is like, do you reckon if you just give them more iron for a long time? Wow, they're not.
Yeah.
Anyway, do you think that you'd maybe see something there?
Potentially, yeah. So there's so certainly 6 months is widely accepted to be a decent, a good duration as well. That certainly wouldn't be over treating, and a lot of that's a common practice in many parts of the world. So potentially, and as you saw, there was a trend towards an increased risk of diarrhea, yeah, so I think I have done some subgroup analysis of, for example, the children who were most adherent to the iron and the MNPs, which didn't seem to show anything statistically significant. But there's also some further work that I'd like to look at, looking at the child's baseline iron status. Because we know that that if you're iron replete, you may have a lot more unbound iron in your colon. So that's an analysis I would like to do next.
Thanks, Andrew. Beautiful data set, admirable undertaking. I was wondering, in your metagenomics data sets, have you aligned to like fungal or viral species as well? And is there anything interesting particularly there?
No, I deleted it. I bioinformatically subtracted anything non-bacterial from the analysis, but yeah. So the aliquots that I used were frozen in the -80, and I used the aliquot that was in the DNA shield, and so everything was sequenced essentially, but I looked at anything non-bacterial.
Cool.
I'm not aware of much relating to iron, but yeah, and in terms of, I think the background parasitic infection is quite low in this area of Bangladesh, as I understand it.
Awesome. Beautiful. Thank you.
Thank you.
Great. Well, we're at 2:00, and I think thanks to the hive mind of the Wednesday seminar, you have another 2 years to go. So that's wanting to hear. But thank you very much for all your hard work. Thank you very much for listening today, and see you next week.